All right, welcome to this last day of this course on basic biology for engineers and big data analysis. Uh, we have been discussing about different omics technologies, some very basic concepts of biology, and also some of the most recent technologies, how one could make use of them, generate big data, and then use that for making sense of the data using machine learning tools. Today we are going to talk about interactomics, how to study protein-protein interactions. This is one of the very crucial biological phenomenon where proteins do not work in isolation. They work in a good team, and that's where they're part of different signal transduction activities when they are like a relay race, when different proteins are going to work together and their activities are going to really determine different type of biological functions. So I'm going to give you first an overview of different methods to study protein-protein interactions, a little bit elaboration on protein microarrays, as well as the label-free biosensors. After this, we will have Dr. Manisha, who is going to give you a hands-on session on protein-protein interaction studies. So what is interactomics? As the name says, you are looking at different technologies, how to utilize that to capture the biomolecular interactions. So interactomics is a study of these biological interactions and their consequences between various proteins as well as other cellular components. Sometimes these interactions could be just protein-protein. It could also be protein DNA, protein carbohydrates, protein lipids, small molecules. So these kind of networks of all such interactions or the interactome that aims to provide better understanding of genome and the proteome functions. Interactomic techniques could be used to identify the function of uncharacterized proteins, because many times from the high throughput instrumentation, you can get a list of large number of proteins, but you have no idea that what these proteins are and what their function is. And most of the time, once you identify the name of the protein, still you are left with determining their function. And to look for uncharacterized protein function, interactomics technology could be very well utilized. Also, we have to use these technologies to define sometime new roles for even already known proteins. And many times we are using interactomics to understand the mechanism which can regulate the protein activity. Of course, these networks of protein interactions are highly crucial to understand various types of biological processes. Most of the physiological process happening, they are because of various binary interactions happening inside the physiological system. These are the complex images which you can see how various protein clusters are interacting, and these nodes and subnodes are very crucial. And that's a lot of bioinformatics and various type of computational tools comes into the play, how to build these kind of protein interaction networks. Also, as on the right hand side, you can see there are several multi-protein complexes which are involved in biological pathways. One could see, in this kind of complex image that how different proteins are going to interplay that important role, which can govern these biological pathways and biological functions. So I'm sure now you are aware and convinced that these protein interactions are very useful. They are dynamic. They're going to interact with wide variety of biomolecules, and their interactions are going to regulate different signal transduction and gene regulation activities. So since you're not too much aware about various type of biological processes, I think it's a good idea to emphasize that these type of protein interactions are very important to govern any kind of events in the cell. Whether we talk about signal transductions, transcription, splicing phenomenon, morphology and growth, motility, metabolism, cell cycle control, translation or DNA replication. These are just few names. All the important biological processes are governed from these kind of protein interactions. So which are the methods to study these type of protein-protein interactions? On one hand, scientists have been using some traditional approaches from long time, which is East2 hybrid, affinity chromatography, and immunoprecipitation. But more recently, there are more high throughput approaches have come forward, which includes the protein microarrays and various type of label-free technologies. Of course, given the time and the focus of the course, I'm not going to talk about each technology in detail. I will hardly cover 2-3 slides to give you some flavor of these technologies. But we would like to give you a little more familiarity of doing this kind of analysis, and that's where we'll have a hands-on session to give you some more illustration for this. So immune precipitation is one of the way to identify these complex interacting partners. So for example, if you have a cell lysate, there is a protein which is going to form the network in a given complex formation, and that if you can pull down using an antibody which is known for that protein which is you are interested in. So then this is that antibody which you are interested in, and you are going to take that antigen which you want to characterize for its partners for protein interaction networks. And now this partner, which is antigen A, which is going to react to this antibody, is also going to come with along with its other interacting partners, which are unknowns for you, and you want to identify those. So you know this antigen which you are interested in, you have raised an antibody against that so that antigen antibody complex could form, and along with that complex, some unknown proteins could also come, which you can do in the pull down assays. Once you have done this, it means you're going to isolate this complex, which contains many proteins, which is your target protein of interest and various interacting partners. Once you have done this, then you'd like to denature them and run them on the denaturing condition in the SDS page to look at which of these proteins are there, which is forming this complex. Then you can go and do the in-gel digestion followed by mass spectrometry to identify which of these proteins are and are there interactors of this protein or not. Again, it's not very easy to answer that are they interactors or not, because like these proteins could be direct interactors, but this protein may be just sticky protein which is coming along with this protein. So we are not sure whether this is actually good interactor or not. So that's why you always see this problem of finding a good interactor which is really known interactor, or sometimes you will have a lot of sticky protein which could just be coming as a part of those complexes. As a result of these limitations, more modern technologies like protein microarrays have come forward, where you have very defined features on the glass slide of the array format, on which you can place or spot these antibodies of interest or purified protein of interest, peptides of interest, or sometimes DNA of interest. And all of this, when you want to find out the interacting partners of a given protein, you can probe that on the chip and look for the signal which all protein is going to show binding or which antibodies is going to go by show binding. And accordingly, you can determine which of these interactors are. Again, each one of these array platform has its own unique features. Each one of this have its own way of chemistry to show the interactions. I'm definitely not going to go in detail right now for this, but of course, if you are interested, we have more lectures which we can share with you so that you can get much broader perspective of how to make use of these technologies to study the interactomics, as well as various type of other biological problems like biomarker discovery based platforms. So we are also using some of these arrays and other technologies to do various projects. In one project, interestingly, which we're working on the colorectal cancer in association with a team from Israel, Tel Aviv University, when we were looking at colorectal cancer gene and the protein signatures. And after doing a lot of work from proteomics, finally what it turned out to be when we did a lot of computational analysis, that most of those proteins which we identified from our project for which is involved in the this disease of colorectal cancer, they were all mapping towards one transcription factor gene. So this became one of the major hub, which is actually governing various type of networks, which is dysregulated in this disease. So these kind of computational analysis could really help us not only to find out which are the changes which are happening, but also which could be the root cause and where all of these activities are going to get governed. And one could find out those key features and then try to target those to do further therapeutic assessment of these cancer. We are also using these array platform for other type of disease biology to understand. In this case, we had collaboration from University of Washington in US, when we have the chips of malaria for the falciparum and vivax, the pathogen which causes malaria. And this project, we had almost 200 patients from Goa, and we looked at how the falciparum versus vivax signals are the same or different. And visibly, you can see the signals are quite different, the falciparum signal as well as the vivax signal. And looking at these, we identified which of those features are quite different, what makes them quite different. Are they different in the non-severe and severe population? Are they different in the young versus adult population? Are they different in different groups living in different cohorts? And all of these information are quite useful to identify the community type of a screening based on the humoral immune response. So this is how some of these technologies have very much practical applications, which could be quite powerful. So when we are talking about microarrays, we are talking about signal detection using some fluorescent dyes like Psi3, Psi5, Lexafluor, other dyes are available. So we have given you some demonstrations, so you are aware of what I'm talking right now. But there is always that challenge that if you are using label-based methods like these kind of microarrays to establish protein-protein interaction or a biomarker discovery, then the tag itself may have certain interference and tag may not be best thing to keep it. So then people have also thought about, can we study biomolecular interactions using label-free platforms? And label-free means like you do not want to tag the protein with a given label like Psi3 or Psi5, but rather you want to utilize the physical properties of these biomolecule itself. Like, the when the two molecules are going to get associated, there will be change in the mass. There will be change in the dielectric property. Can we use those properties and try to measure the signal just in the native condition rather than modifying them chemically by using the fluorescent tags? So label-based methods are very popular because of the kind of instrumentation required and the signals are very strong. So fluorescent method, radioisotopes, horseradish peroxidase, some of these are the ways to look for the label-free detection based work. But label-free is also coming up and many technologies have come forward, which is based on surface plasmon resonance, carbon nanotubes, biolayer interferometry. Many methods are there, which is being used for studying these complexes and the biomolecular interactions in label-free manner. So label-based, I have given you already some flavor. Again, just an overview slide for the label-free techniques. One could use carbon nanotubes as multicolor ribbon labels. One could use planar waveguide arrays, nanowired sensor arrays, spectral reflectance imaging biosensors, nanohole arrays, surface plasmon resonance. I have talked briefly in the past. Some of these are the ways to think about how to study these kind of biomolecular interactions in the label-free manner. Let me just give you one example in some detail: nanowired sensor arrays. Think about these type of nanowires, which is having an antibody of interest. Let's take the example of currently SARS-CoV-2, which is everybody wants to learn about it, right? So from this virus, if you can identify the proteins, and if this virus is, let's say, adhered in our human body, uh, if there are some sensors which contains antibody against this viral protein. Now, if this viral protein comes out and attached to the antibody, which is on the nanowired sensor arrays, then you will see a change in the conductance because of that antigen antibody binding happening. Antibodies are very specific, derived from the specific epitopes of the protein, which is specific for that given protein of your interest. This example has not been tried out in the COVID yet. Of course, that opens up opportunity for the participants here to try out something new, but it has been used. Even there was a PNAS paper 2005 where they actually showed that this type of technology could be utilized for virus detection on the airport. And they showed some virus outbreak at that time that it could be detected on the metal detectors if you have these nanowires sensor arrays. This could be utilized to find out if the virus is binding to the antibody, viral protein is binding to the antibody, and then accordingly that there is a change in the conductance. Very quick way of measurement, and that could actually screen those potential people who are having these contamination, and then they can be kept separately for further test. So all of these are, I'm sure giving you some flavor how practically they can be utilized, how well they are powerful technologies to make sense of them and bring them into our actual practices. So what we are talking right now to you to study these proteins and their interactions and their various properties to build the layer by layer information, which is very important if you want to understand any life science system, any biological system, of course, human system, where these proteins are localized, how these proteins interact, which are the substrates for these proteins, what are the assay conditions which we can develop to understand these proteins. So you can't just study them in isolation. You have to look for making these layers of information and then try to understand a system which can give you much powerful information, much beyond just looking at the single molecule. I hope this gives you flavor that studying the proteomics, the complex protein molecules, and their networks of interactions are very powerful and biologically very relevant. Also, it is having lot of application which can have applications in different domains of life. I'll stop here for this part. Let me now invite Dr. Manisha to give you a session on hands-on session on the protein-protein interactions. So she will give you some overview and then followed by a hands-on session.
Actions. So the contents of this session are importance of protein-protein interact. So the contents of this session are importance of protein-protein interaction analysis. So we will try to understand like why is it important to understand the protein-protein interactions, and also the binding site analysis, the various docking methods, also the concepts behind rigid and flexible docking. Then the further on moving ahead, we will try to see the different softwares available, and we will be taking a hands-on session on that. And also finally, we will have a look at Chimera for visualization. So the importance of protein-protein interaction studies is huge, and so the main reason why we have to carry out protein-protein interaction studies is for predicting like how two proteins would interact with each other. For example, if you consider this particular protein A and this protein as B, and you can see this is how the shape complementarity looks like, and this is the shape complementarity of protein B and how they form a complex. So the applications of protein-protein interactions are quaternary structure prediction. For example, if we look at the blood coagulation mechanism, so we have two kind of complexes, two kinds of ways by which this mechanism takes place in the body. So one is the extrinsic teinase complex and other is the intrinsic teinase complex. In case of extrinsic teinase complex, so we have factor 7A, which interacts with the tissue factor, and then the factor 10, it gets activated to factor 10A. So now this particular mechanism, this is the extrinsic teinase complex. So this is how the blood coagulation pathway or the it takes place in the body. So we can see how the proteins they come together and they form a complex, and then only these pathways they regulate in the body. The proteins are very densely packed inside the cell. So almost 20 to 30% of the total volume of inside the cell are proteins. So we can see how the proteins they form complex with each other. So definitely when there are so many proteins inside the cell, it is not that one protein will take up only one function in many of the cases. There will be several proteins, they will group together and form a complex, and then they will take up one function in the body. Another importance that needs to be considered is that many biological processes, as discussed, are controlled by protein-protein interactions. So here we can see this is a protein interaction network in case of cell signal transduction. So these pathways such as signal transduction, transport, and cellular motion, these are highly dependent on several protein complexes, which regulates them. Further moving ahead, one of the important topics that need to be considered in case of protein interaction analysis is the binding site analysis. So the proteins, they can come in contact with each other not only in one patch, but two or more patches. If we can see the how protein is, two proteins are interacting with each other, so we can see that there are several modes in which two proteins can bind to one another. So this is always a difficult task in case of protein-protein docking studies to actually determine which is the best binding mode. So if we have some information from the crystal structures that can give us proper information on the binding sites of the particular proteins, then it becomes much easier to determine which is the best complex that can be considered. Some residues, they have a very high propensity to be in the binding site. Usually, be it polar or non-polar residues, they have their own characteristics and which makes them to be placed in the different binding sites. Now, the residues in the protein-protein interfaces, they have been observed to be highly conserved compared to other residues in the protein. And many of the residues, they not only contribute to the interaction between the two proteins, but also to the binding energetics. There are several docking methods. First is wherein we all know about the protein ligand docking, and then protein-protein docking. So one of the similarity between protein ligand docking and protein-protein docking is that both the docking approaches, they look for the best conformations and the best lowest binding free energy. So the differences the two have are first, in case of protein docking, so the surfaces between the proteins wherein they interact, they are large and they are flat. Mostly the residues, they are conserved and they have a hydrophobic core. And in case of binding energetics, it's usually dominated by the geometry of the protein and the hydrophobicity of the residues present in this interface. Protein flexibility is important in case of protein-protein interactions, and which is contributed mostly by the side chains and the backbones. Now, coming to the concepts behind rigid and flexible docking. So rigid docking is wherein we are considering more about the shape complementarity and the electrostatic complementarity. So in rigid docking, so the protein, it will be in its particular conformation throughout the entire experiment. It won't change its conformation. So the search methods include exhaustive algorithm and FFT. In case of flexible docking, this is actually the one step ahead of the rigid docking. Here, what we are considering is that the protein would change its conformation during whenever the ligand is binding to the binding site of the protein. So this is basically contributed by the sidechain rotamer libraries, which are the various libraries of various rotations in which the sidechains can rotate. Then Monte Carlo algorithms and genetic algorithms are widely used in case of flexible docking. So as we know that these kind of approaches, the in silico approaches, have widely reduced the time of drug in case of drug designing. So the for rigid docking, which is the main aspect in case of high throughput virtual screening. So there are several thousands of small molecules which are being filtered, and finally we have only 4 to 5 small molecules which are actually considered and found to be of interest in the studies. There are several softwares which are available online for protein-protein docking studies. These few of the softwares are available online, and few softwares can be downloaded and installed in the either laptops or desktops, which can be used later. Few softwares such as ClusPro, PatchDoc, Piper, HatDoc, HDoc. So these are very widely used by the bioinformatics community. So here in this session, we will talk about the software ClusPro. So now we will just go and try to do it online. So if you just type in ClusPro, so ClusPro is a software which is maintained by the Boston University. So we have two options. Either we can log in, we can open our own account and we can log in so that we can trace back all the docking studies that we have done using ClusPro, or we can just opt to use ClusPro without account. So it is usually protein-protein docking is much more easier, because what we are doing here is mostly blind docking. So it is easier because we are not specifying any grid. So that grid means a space wherein the particular ligand should go and bind. We are not specifying any region. Rather, we are allowing it to go for blind docking. So it will actually take the ligand protein throughout the entire target protein, around the entire target protein, and try to dock it at different places and give us different complexes. And we have to choose the best possible cluster based on our previous information. So docking, sorry. So what we'll do is, we will just give a job name here. Let us just call it as docking protein. And we'll make this as GPU so that it is done faster. We'll go to receptor. We will upload a receptor protein, which in this case is the tissue factor, which if you remember in the blood coagulation mechanism, I showed that factor 7A and tissue factor, they both interact. That is a complex protein. They both, that is a complex they formed by both the proteins, and then only factor 10 gets activated. So we will actually take those two proteins. So you can take the PDBs from RCSB. So I'll just show you once again from where we can take the PDBs. We can just type in RCSB, which will take us to the protein data bank. So here, if we put in as tissue factor, so these are structures for tissue factors. Sorry. So I think tissue factor human. So you can see here the several structures are present. This is the extracellular domain of human tissue factor. Okay, so these kind of tissue factor structures we can take, and so the only thing that we should always check before taking a protein is that so it should not be a chimeric protein, first thing, so it should not be a mutated protein. And the second thing that we should check is that is the organism from hosts and not the organism. About human proteins, then we should look for the organism. If we are talking about mouse proteins, then we should take from mus musculus. So that way, and also we should check the resolution. So here you can see that it has 2.4 Armstrong resolution, so it can be taken. But if it is like 3.2, so we should always try to prefer less than 2.5. So now if we actually click here and want to know more about the protein, so all these informations can be obtained about the protein. The method in which, if you remember in the last session, we discussed about the different techniques which are used to determine the structure, three-dimensional structure of proteins. So one of the method is X-ray diffraction. So here, this method has been used to determine the structure of the tissue factor. Then, so this is human tissue factor, A and B chains both are there. So it is 219 amino acid sequence length, 0 mutation. So this is the information that we can get from here. And also, it is important for us whenever we are taking any protein for any bioinformatics studies, we should know further about the protein, like what are the inferences that the team which has determined the structure, the scientists who have determined the structure, the research papers that they have published on that particular protein. So we should go through those papers so that we get more information about the protein regarding its structure, regarding its important domains and the binding site regions, the catalytic, if it is an enzyme, so where does the cofactor bind, and all those information should be obtained. So if I just open here, so this is where the link of the paper is given. So if you go through this paper, we will get all information about the crystal structure, starting from what are the binding site residues and everything. So this is the paper, the mechanism of an inhibitory antibody on tissue factor. So this is the paper. So this is how we have to do a basic research on the protein which we are considering for our biological studies, for our protein-protein interaction studies. So now coming back to ClusPro. So here, as I have uploaded already the tissue factor protein, I'll upload sorry, I did a mistake. So I should have uploaded the factor 7A here as the receptor, and here I should have uploaded the tissue factor as the ligand. So I'll just upload this once again. Okay, so I have uploaded factor 7A and then tissue factor, and then there are several options here. If you plan to do some interaction studies on antibody, And then if you want to modify a structure, so all those options are given. So we can just agree and say doc. So if you see in the screen, so this is the ClassPro after we have clicked on doc. So these are the jobs that are running on the ClassPro server. And here, so all the people, all the scholars or the researchers throughout the world, they can access this ClassPro program, ClassPro server, and they can give their protein docking studies, upload their protein docking studies here. And so this is a freely available software which everyone can use. And if you can see here, our study, which is the docking protein. So our job, this is already uploaded. So we can keep checking the status. So now it is saying that it is processing the PDB files, because before running on the supercomputer, the structure will be cleaned up, the format, everything would be arranged in the format that is required. This is just like we prepare the protein and the ligands before going for docking studies, molecular docking studies, wherein we remove the water molecules, clean up the protein structure, add charges to the ligand. So all these kind of cleanup and everything processing also needs to be done before carrying out a protein docking experiment. So now this job will take some time. So we will not wait till then. So I already have analyzed this file, and therefore I will just go back to the PPT. So if you can see here, once the job is completed, so it will come under my finished jobs, and we can see that our job, which is suppose docking, so it got over and it will say finished. Now this server will keep the information about all the analysis only till 3 days, after which it will delete all the information from the servers. So it is suggested that if we are doing any kind of protein docking studies, and which is uploaded in ClassPro, then we should download the files as soon as possible after the status shows finished. So this is how the result page looks like. So if here, this is the job details is given, and we can actually visualize the model scores also. If we have any information about the structure of the protein and how the complex would form, would look like, and any information about the complex, then we can choose to look for electrostatic favored or hydrophobic favored, or van der Waals and electrostatic interactions. But if we do not have any information, so we can go for balanced. So these are the various clusters of complexes that is being shown here. So we have to basically look for each complex and try to make sense of what is our expected result. So it is always better to have some information about the proteins, the two proteins that we are docking, so that we can have some idea beforehand that that this is the binding site wherein the protein is the ligand protein is supposed to bind, and then we can shortlist and go for a energy calculation using other commercial softwares, and further go for calculation of energy and choosing the best pose from there. We have an option of downloading these files, these complexes. So if you just click on 0 or 2, any of this, so this model will get downloaded in the PDB file format, which we can further save in our system and analyze using other softwares. So now we will go for the visualization of the complex that we have obtained. So Chimera is a very widely used visualization software, so which is very commonly used and which really gives very high resolution pictures. So this is how the Chimera software looks like. So this is freely available, and the software can be downloaded and then installed in either laptop or desktop. So in the Chimera workspace, here whatever the files that you have been working on, for example, the PDB files, so those can be uploaded in the workspace, and it also gets saved. So recently I have worked on all these files and it got saved. So I do not have to take the pain of browsing the files and bringing it here. So I can just select it from here. So now, suppose I want to check the complex file which I have got from ClusPro. So this is the file. The file got uploaded, and this is the protein complex. So here we have the heavy chain, light chain, and the tissue factor. So heavy chain and light chain were in factor 7a, and tissue factor was the protein that we wanted to dock. So I'll just make the background white. Then we can color each chain so that it will be easy for us to differentiate. I can just go to color. So what I did was went to chain and selected heavy, which will select the heavy chain. I went to color under actions and then just say green. So the heavy chain got green in color. Then I will select the light chain and make it blue in color. Then I went to the tissue factor and made it magenta. Okay, so now you can see that all the three chains are different in color. Or both the proteins are different in color. We have only two proteins here. The heavy and light chain belong to factor 7a, and the tissue factor is bound to it. So the next thing what I would do is I would want to look for some interactions. So for that, I'll go to tools, I'll go to structure analysis and say find hydrogen bonds. Now this will give me an option of choosing whether I want to show intramolecule hydrogen bonds or intra-residue hydrogen bonds. So all these options will be there. So I'll just select intramolecule hydrogen bonds because I'm interested in only the interactions between the three chains. So I'll just say apply. So now you can see here the different hydrogen bonds are being shown. So more than the intramolecule hydrogen bonds, we are interested to know more about the intermolecule hydrogen bonds. That is bonds like this one, right? If you just take the mouse over here, so it shows that the interaction is between tryptophan 45 and phenylalanine 129F. Now, if I want to select these two residues, I will just go to tools. I will ask for the sequence. So I have, because there are three chains, so it is showing as chain L, light chain, heavy chain, and tissue factor. first I will go to the heavy chain. So, sorry. So we know that the residue is phenylalanine 129F. So let us look for that. This is phenylalanine 129F. We have selected the residue. Then we'll go to tools, actions, atoms, show. Okay, so now you can see this particular atom is being shown, right? Because whatever we are seeing here is the ribbon form. Okay, so when I said selected the residue and I'm asking to show it as stick atoms and stick stick model, so it is showing me that particular atom, which is the residue. So that residue is shown in the stick format. Then what I will do is I'll go to the next sequence, which is tissue factor. If I say show, now this particular residue is tryptophan 45 of tissue factor. So this is the residue. I'll go to action, show. So you can see this one, this format. So it has come in the ball and stick format. So now, so this is what we can see in Chimera. Different kind of analysis can also be done here. So this is one thing. And the second thing that we can do here is, if I'm selecting this particular chain, heavy chain, suppose, and then I want this to look not like ribbon, but as surface, I'll just say show. So now this looks like a surface. If I want some transparency, I can just go to action, go to surface, go to transparency and say suppose 70%. So now the structure has become a bit transparent. So this kind of images which are required for publication, so those images can be made here. We can also type in the residue names and distance between the bonds. Everything can be done here. Another feature of this software is, so I'll just clean up the workspace. So suppose I have two proteins. So I have the tissue factor which I wanted to dock. Now this is only the tissue factor. Okay, this is only the tissue factor protein. Now I also want to see that the protein, receptor protein, which is having the heavy and light chain. Okay, after it got docked with the tissue factor, so it has formed a complex. Now I want to see that the wild type only tissue factor and the tissue factor which is present in the complex. So how different have they become in their orientation? So is there any orientation difference between the two proteins? So let us just upload the complex. So you can see the complex is here. So if I just make the background white again, then I will go to tools. I'll just go to structure comparison, go to matchmaker. Then I can select one protein on this side, left hand side as the reference structure, the other protein as the structure to match. So I'm selecting tissue factor, the only tissue factor protein, and then the complex protein, and then I say apply. So if I want to also see the sequence alignment, I can go for pairwise alignment. So now you can see here that the two proteins had got overlapped. Okay, so you can see this is the light chain and the tissue factor of the complex and the tissue factor which we later uploaded here. So these both have got overlapped. So this is how this kind of superimpositions of structures also can be done using Chimera. So we can color these chains as different in different colors. So then we can actually see the difference. So then in terms of sequence alignment also, we can see if there is any difference among the two sequences once we overlap it, then also that can be seen here. So this is the sequence alignment which we did in our previous session. We did the cluster omega. So here what we are doing is the structural superimposition. We want to see how similar are the two protein conformation and orientation. So now I think this is all about Chimera. So this is a very easy software which you can download, free software you can download, install in your laptops and explore it further. The second software is Pymol, which I think most of you might have used or at least heard about. So in this software, so I have uploaded one protein which is ACE2. So that is a very common protein which most of us are hearing in COVID. So if you see the screen, so this is ACE2 protein, and this particular protein is actually bound to hydroxychloroquine. Now we want to, if we want to know further, so if I see this protein like this, so I cannot get any information. So I will just go to all preset ligands. So what it has done is it has shown me the ligands present in the protein. So this is the hydroxychloroquine drug, which is docked here in ACE2. Okay, and these are the these bars which we can see here, yellow color. So these are the hydrogen bond interactions. So just like we saw in Chimera, that was between two molecules, between two chains. This is between the ligand and the protein amino acid. So now here also, if I want to see what is this particular residue which is showing interaction to hydroxychloroquine, I will just click here, select it, and just say label the residue. So I know that it is aspartate 367. I can also move these labels. So now it is clear. So aspartate 367 is one of the residues that this particular ligand is binding to or showing interaction. And this interaction is between, this red colors are the oxygen, these whites are the hydrogen, and these red are the oxygen. So this is a hydrogen bond interaction. So these kind of structure visualizations can be done, and several other features are also there that can be explored. So these softwares are freely available online, which can be tried. So I think I have come to the end of the session. So if there are any queries or questions that you want to ask, so I'll be very happy to answer. Thank you.
All right. Thank you, Sanjeeva.